Well, good afternoon, everyone. My name is Liz Crane. I'm the Associate Director and Head of the In Vitro Pharmacology Group within Antibody Discovery Services at Alloy Therapeutics. And today I'll be sharing a little bit with you about how Alloy is redefining bispecific discovery through our integrated design and functional screening workflow, and specifically highlighting some work that we've done to find safer, more effective T-cell engagers. So Alloy might be best known for our humanized mouse models for antibody discovery, but Alloy also has an integrated bispecific discovery workflow for design and functional screening. And we have the expertise and experience to not only discover bispecifics, but do it in ways that span a variety of mechanistic areas of need and biologic requirements. So specifically, we have the capabilities of designing and testing bispecifics for use as cell engagers, like the T-cell engagers that I will be highlighting today. For dual screening blockers, or dual signaling blockers, for example, cytokine blockers. We can also design bispecifics to use as biparatopic antibodies for for ADCs, and also for biparatopic receptor modulators, whether you're trying to stimulate or suppress your receptor signaling. So our capabilities really for bispecific discovery, engineering, and functional testing, we really kind of been into these three pillars of design, scale, or production, and then selection based on characterization and functional screening. So for design, Alloy can work with any format of antibody, and we routinely use any format or multiple formats as building blocks to really build the bispecific, purpose-built antibodies. For scale and production, we have capabilities for high-throughput production in 96-well formats, all the way up to gram-scale productions in 24 flask-based format. And all of our antibodies go through a robust purity and QC screening. We then move molecules into our third pillar, which is the selection pillar, where we identify leads, again, based on characterization and a whole host of functional assays, really, again, tailored to the biology needs for the program and the mechanism of action. Where do these building blocks come from? So again, at Alloy, you can take advantage of the wonderful humanized mouse models that we are continuing to develop and push the boundaries of, including our CLC models. You can do de novo discovery using our VHH discovery and phage library screening systems. Or if you want to kind of jumpstart your program and save a little bit of time, you can also use off-the-shelf building blocks through our Foundry division. Or you can bring your own sequences, and Alloy will work with you to potentially even make them better, but also use them to design the molecules that fit your needs. So for Foundry, the options, again, if you're wanting to jumpstart your project and save a little bit of time without going through de novo discovery, Foundry has these optimized off-the-shelf molecules that you can use as your building blocks. All of these molecules cover a diverse panel of human diseases and targets, and they come with robust characterization packages that you can look through that include a lot of characterization data, binding, kinetics, and preliminary functional testing. Molecules that are available, again, cover a wide variety of biological use cases, mechanisms of action, again, cell engagers like the CD3 antibodies that I will be highlighting today, but then also target modulation, delivery for use cases in ADCs, or to deliver your therapeutics to different disease types of interest. And speaking of CD3 antibodies in use in the T-cell engagers that I will be highlighting today, again, Foundry offers this diverse affinity variance to really dial in your use case. So we have an array of both SP34 and UCHT1 antibodies that cover a variety of variant space, not only in the monoclonal format, but then when converted to scFv FC formats, you can see the high affinity UCHT1 clones and the mid to low affinity SP34 clones that are available. So now to jump into a case study that really highlights our end-to-end bispecific discovery capabilities, specifically looking in this case, into a T-cell engager. So a couple of the big well-known problems when looking at T-cell engagers are not only ensuring that you have very potent molecules that can induce the killing that you're looking for, but also then taking into account or mitigating either off-target effects or risks of cytokine release syndrome. And so to cover both areas of these concerns, we leverage the building blocks with the affinity variants that we have available to us for the CD3 arm of our T-cell engager. And then we also engineer these formats to improve the therapeutic window, the potency, while again mitigating these off-target effects. And then we finish out with our functional screening, so really taking advantage of all of our functional testing capabilities to funnel down and make sure that we end up with the best molecules possible at the end of the day. So as I said, we are really employing our ability to test a variety of affinity variants within this program, and we're using both affinity variants on the CD3 arm as well as the targeting arm. So we're modulating both sides of the equation. On the CD3 arm, we're looking to, again, be able to activate the T-cell population while lowering the risk of a cytokine release syndrome and those off-target effects. And then on the targeting arm side of things, we're wanting to make sure that we dial in the affinity enough to make sure that we have enough targeting and potency while mitigating any sort of targeting for normal cell populations or any sort of low antigen expressing cell populations. And then taken together, we combine both arms of affinity variants to create this nice matrix of molecules to then go into the full screening funnel. So taken together for this program, we ended up using 5 CD3 clones for the CD3 arm, 10 targeting arms for the targeting arms side of things. Multiple formats were used and explored in this program, and then at the end of the day, we ended up with a broad panel of bispecifics to then go into the scale production and screening parts of the program. So problems can arise even with great planning, and we actually ran into a problem kind of immediately upon entering that scale production phase of the project. So we identified a clipping issue in one of our critical clones that was really in that mid-affinity range area. So liability analysis hadn't shown any potential cleavage sites within this molecule, but during our QC stages, this clipping was seen. So in order to address this and salvage that clone for this program, we were able to identify the cleavage site through mass spec after a little bit of investigation. Then we were able to rapidly move into creating a variant panel using our in-house AI ML capabilities. And within weeks, we were able to then produce and characterize a panel of variants and identify a number of potential clones that showed no clipping and this loss of clipping liability. Not only that, through our full screening and characterization capabilities, we showed that not only did we lose this clipping phenotype, but we also maintained the desired affinity for these variants in order to salvage this clone for this project. So then moving from the production side of things, then we went into our first functional assay screen for this program. And as this is a T-cell engager program, we really wanted to tailor our assays to the specific biology needed for that molecule. And so to do this, we used a T-cell dependent cellular cytotoxicity assay, or a TDCC assay. And this is a type of assay where you put in your target expressing cell line, T cells, and then your TCE antibodies. And our readouts included measuring cell death along with multiplexing for cytokine release at the end of the day. Now, we didn't just want to throw any sort of cell line into this assay. We really wanted to tailor this assay to again, being as biologically relevant to the disease as possible. So we screened a variety of immortalized cell lines, and we quantified their target copy number to line up with the expression levels on disease-relevant human cell populations. And through this screening, we were able to identify and choose 2 cell lines that corresponded with high expression and low target expression relative to disease-relevant human cell populations. And so here we established a high-expressing and a low-expressing high-throughput assay system to initially screen all of these bispecific molecules. And what we saw was that a group of lead molecules not only showed as good as or better potency than our clinical benchmark, but they also showed lesser killing activity in our low target expressing model. So in the circles at the top, you can see our bispecific molecules are as potent at killing cells as the benchmark. But then in the squares at the bottom, there was actually this reduced level of killing seen in the low target expressing cell lines. Again, suggesting kind of an improvement in our risk factor for killing of normal healthy cells in this situation. And so this therapeutic window, we showed that a number of our molecules had an improved therapeutic window up to 10 times greater than our benchmark antibody. So we were able to show potent cell killing, but what about cytokine release? And we are actually pleased to see that in some of our lead molecules that showed equivalent cell killing potency, we actually saw quite a significant reduction in expression of interferon gamma in these models. So at a concentration where equivalent cell killing was seen, we actually saw a 3.5 times reduction in the level of interferon gamma released. This was in the high expressing model. So again, we're seeing either improved or similar potency to the benchmark with an improved safety profile. So then we wanted to move further down our screening into a cell-based assay model that incorporated, again, more disease-relevant, physiologically relevant cell types. So this moved into a primary cell-based assay where we used primary disease-relevant cells mixed with our bispecific antibodies and looked at killing of disease-relevant cell populations with spectral flow cytometry, and again, multiplexed with cytokine readout. And on the right there, you can see that our lead molecules again showed a higher potency of killing in our high-expressing disease-relevant cells, while either showing a reduced or equivalent potency on mid or low expressing cell populations. We were able to induce T-cell activation while not augmenting cytokine expression in any way. So again, this just highlights our ability to screen affinity variants across a number of bispecifics. We were able to identify leads moving forward. We identified an optimal format for these bispecifics, the best combination of both targeting and CD3 arms that gave us the optimum and the desired profile at the end of the day. So we're a relatively flexible team. We move quickly, and we can really tailor our workflows and our screening funnels and all of our assays to the needs of the program. We really try to work with our partners and make sure that all of the desired phenotypes in their molecules are pulled through, and we really screen them and test them in the most appropriate systems possible. And if you are interested in our entire workflow, we can move all the way through from de novo discovery through production and screening in a matter of weeks. We have a number of tools available to us outside of just our wonderful mice, and we can work with you throughout the entire process or at any of the stage, again, if you have your own molecules or if you want to use off-the-shelf building blocks to really accelerate your projects.